Good afternoon, everyone. I'm glad to be here with you today. I'm Ppeum Han from Chung-Ang University. Thank you for this opportunity in New Agro-Food Research Institute of Chung-Ang University seminar. Today, I will presenting on development of novel feeding strategies to improve egg quality and healthiness in aged laying hen using nutrigenomic approaches. This is an overview of today's presentation. First, I will talk about the background, followed by experiment 1, 2, 3, and 4, and conclude the presentation with a conclusion. Chickens are one of the most common and widely distributed domesticated animals, broadly categorized into breeder, broiler, and layhen. First, breeder are chickens that lay fertilized eggs for hatching chicks for the purpose of breeding. Second, broiler chickens raised for meat production, typically slaughtered after about 5 to 6 weeks of age. Third, laying hen raised for egg production, starting to lay eggs around 20 weeks of age and continuing for about 1 year until they reach 80 weeks of age. The primary goal of laying hen production is to yield a greater quantity of high-quality eggs. In my research, I have focused on laying hen among these three types of chickens. Therefore, to achieve a greater quantity of higher quality eggs, the majority of research on laying hens is focused on enhancing productive performance and egg quality. Productive performance is assessed through metrics such as weight of lay, egg weight, feed conversion ratio, whereas egg quality includes eggshell color, eggshell strength, and etc. However, the decline in productive performance and egg quality, which are important indicators of laying hen, is especially a problem in aged laying hens. Consequently, previous studies on aged laying hen have made efforts to improve productive performance and egg quality. However, results are inconsistent. One of the reasons the results were inconsistent was most previous experiments with aged laying hens were feeding experiments based on trial and error method. Recognizing the limitation of the traditional trial and error method, we believe that it was essential to identify the precise factors and mechanisms behind the decline in productive performance and air quality. However, molecular biological research related to productive performance and air quality has been notably deficient up to the present. Therefore, we aim to elucidate the mechanism for improving productive performance and egg quality through a molecular biological research, specifically employing a nutrigenomic approach. My focus involved utilizing transcriptomics to identify key factors in this process. To compare the expression of genes, the most commonly used method is to measure mRNA, and transcriptomics is the study of transcriptome, which is a set of all mRNAs. In other words, transcriptomics can be defined as a study that deals with life phenomena at the mRNA level. Among the methods for analyzing transcriptomics, RNA sequencing involves converting mRNA into cDNA and subsequently utilizing next-generation sequencing to analyze all the nucleotide sequence present in a sample. Therefore, I conducted transcriptome analysis using RNA sequencing as a means to investigate gene expression patterns. This is the general outline of the experiment. We investigate to identify the region of egg quality decline through analysis of transcriptome in tissue related to nutritional metabolism in aged laying hen with different egg quality, focusing on eggshell color and eggshell strength. After identifying the factors that reduce egg quality through these transcriptome results, we ultimately conduct research to develop a novel feed that improves the healthiness and egg quality of laying hens. Experiment 1 and 2 aim to identify the reasons of egg quality deterioration using transcriptomics in aged laying hens with different egg quality. Egg quality can be broadly divided into 5 categories. There are eggshell color, eggshell strength, eggshell thickness, whole unit, and egg yolk color. So, let me tell you why eggshell color and eggshell strength are important in eggs. If eggshell color is not uniform or the eggs are malformed, it is difficult to motivate consumers to purchase. Especially in the case of brown eggs, eggshell color is an indicator of consumer preference. Additionally, eggs have a long distribution process, and eggs often break or crack during the transportation process. When eggs break like this, the level of bacterial infection through the pores increases, so it may become the cause of microbial infection. Therefore, research is being conducted to produce eggs with stronger eggshell strength and thicker eggshell thickness. In particular, eggshell color and eggshell strength are a major problem in aged laying hens. As the chickens age, the egg weight increases, but the amount of calcium and the amount of pigment are the same. This is because as laying hen age, their ability to utilize calcium and produce pigments that produce eggshell color decrease. Therefore, as chicken aged, eggshell color is paler and eggshell strength is weaker. Due to this problem, aged laying hens are being culled. Therefore, resolving this problem is very important. Transcriptome studies have been conducted to resolve this problem of poor quality in aged laying hens. However, most previous studies were related to uterus, the tissue that produces eggs. However, as you can see, in addition to the uterus of laying hens, the liver is a tissue that plays a very important role. Liver is not only the central tissue of nutritional metabolism, but also a tissue that performs many important functions such as digestion, storage, synthesis, and detoxification. Although liver is an important tissue, studies related to egg quality have focused only the uterus. Therefore, currently, the transcriptome research on liver to improve the egg quality of aged laying hens is very insufficient, and this can be said to be a reason why this study was conducted as a study to improve the egg quality of aged laying hens. Therefore, we conducted a hepatic transcriptome studies to identify the cause of egg quality decline in aged laying hens. The purpose of experiment 1 was to identify potential factors affecting brown eggshell coloration in aged laying hens at the hepatic transcriptomic level. And the purpose of experiment 2 was to investigate potential factors affecting eggshell strength in aged laying hens at the hepatic transcriptomic level. Experiment 1 is transcriptomic analysis of the liver in aged laying hens with different intensity of brown eggshell color. This is the method used in experiment 1 to select laying hens with different eggshell colors. After tracking the eggshell color for 3 weeks using 500 highline brown aged laying hens with 92 weeks of age. After final selection of 6 laying hens with darkest eggshell color and 6 laying hens with the lightest eggshell color, and the liver tissue was sampled. To explain the transcriptome analysis process in more detail, first, total RNA is extracted from the liver sample in the experiment, and cDNA is synthesized, and the adapter are added, and amplification is performed. The amplified product was sequenced, and bioinformatics analysis was performed using the raw data from the sequencing. Afterwards, after checking the quality of sequence data, after removing the previously attached adapter and filtering out reads with low quality scores, we mapped to the chicken genomes by quantifying the expression level of each gene. Expected differentially expressed genes were discovered. Functional analysis were performed using a previously known biological mechanism database. These are the results of experiment 1 to investigate transcriptome changes in laying hens with different eggshell colors. The x-axis of each graph represents groups of laying hens with differences in eggshell color, and y-axis is the numerical value of each item. There was no significant difference in eggshell strength between laying hens with dark eggshell color and those with light eggshell color. On the other hand, in terms of eggshell color, laying hens with dark eggshell color showed darker eggshell color than laying hens with light eggshell color. The colorimeter result also showed significant differences in lightness and redness value, which are most meaningful in eggshell color. Laying hens with dark eggshell colors had lower lightness and higher redness value than those with lighter eggshell colors. Therefore, this confirmed that the selection of laying hens with different eggshell colors were successful. This is the hepatic transcriptomic result. The figure on the left is a volcano plot, and when a differentially expressed gene called DEG was discovered with a p-value of less than 0.01, there were 290 DEG in laying hens with dark eggshell color as compared to those with light eggshell color. The figure on your right is the multi-dimensional scaling plot. As you can see, two groups were not clearly differentiated in terms of gene expression differences in liver tissue. These results suggest that eggshell color differences are not due to differences in the expression of a large number of genes, but rather to differences in the expression of some genes. The figure you see is showing the protein and protein interaction. It was confirmed that the network of unregulated genes in the liver had a stronger influence on the degree of brown eggshell color. Additionally, most of the nodes in a second large hop contained genes related to immune response. genes related to the ability to regenerate and repair the liver were regulated in lineage with dark eggshell color. This is the result of the cake pathway hepatic transcriptome according to different eggshell color. Overall, lineage with dark eggshell colors has stronger immune responses, oxidative stress, and had a developed vitamin A metabolism and had an excellent ability to detoxify ammonia. In addition, it was confirmed that the gene expression associated with fatty liver disease is increased in aged hens with light brown eggshell. Considering this, it was predicted that fatty liver disease, one of the factors that reduces eggshell color in hens. Experiment 2 is transcriptomic analysis of the liver in aged hens with different eggshell strengths. This is the method of experiment 2, which selected hens with different eggshell strengths. After tracking eggshell strengths for 3 weeks using 500 highline brown aged hens with 92 weeks of age, after final selection of 6 hens with strongest eggshell strengths and 6 hens with the weakest eggshell strengths, and then liver tissue was sampled. RNA sequencing was performed on this liver tissue sample. These are the results of experiment 2 to investigate transcriptome changes in hens with different eggshell strengths. It was confirmed that hens with a strong eggshell strength had significantly higher eggshell strength than those with weak eggshell strength. Eggshell thickness also showed similar result. However, there was no significant difference in eggshell color between the two groups. As a result, it was confirmed that the selection of hens with different eggshell strengths was successful. The figure on the left is a volcano plot when differentially expressed genes were selected based on FDR value of less than 0.05 and absolute log F value of 1. Hens with weak eggshell strengths had 2084 DGs as compared to hens with strong eggshell strengths. The figure on the right is an MDS plot showing a clear distinction between two groups. It can be seen that the expression patterns of hens with strong eggshell strengths and those weak eggshell strengths are clearly differentiated. The figure on the left is a figure showing the protein and protein interaction. It was shown that various genes interact with each other. In more detail about which genes played an important role, using 5 centrality calculation method, we discovered that 3 common hop genes. All of these genes were regulated in weak eggshell strengths. First, the growth factor receptor bound protein 2 genes was identified as a candidate gene for fatty liver disease in a previous paper and was found to be increased under oxidative stress. Second, overtransferrin genes is known as an egg white related gene, but more research is needed to determine exactly how eggshell strengths, egg white, interact with each other. Moreover, previous studies have shown that Overtransferring genes increase inflammation situation. Lastly, CBL proto-oncogene is a gene related to bone resorption. Dietary calcium is absorbed into the small intestine and moves through the bloodstream, causing calcium to be stored in the bone, and the calcium may be deposited in the eggshell. Previous studies have shown that overexpression of CBL genes reduce bone resorption. From this, it can be inferred that laying hens with weak eggshell strength have a reduced bone resorption rate. This is the result of KEG pathway hepatic transcriptome according to different eggshell strengths. Overall, laying hens with strong eggshell strength has stronger calcium metabolism, vitamin C pathways in the liver. Additionally, glycolysis pathway is increased, but metabolic pathway related to gluconeogenesis, fatty acid synthesis, and fatty acid oxidation pathways is decreased in laying hens with strong eggshell strength. In addition, it was confirmed the gene expression associated with fatty liver disease is increasing aged laying hens with weak eggshell strength. Considering this, it was predicted that fatty liver disease is one of the factors that reduces eggshell strength in laying hens. As a result of experiment 1 and 2, it was confirmed that gene expression associated with fatty liver disease is increased in aged laying hens with poor egg quality, including light brown eggshell, weak eggshell strength. Therefore, I thought that if we could prevent fatty liver disease, we could improve the egg quality of aged laying hens. In order to improve the decline in eggshell color and eggshell strength, it is believed that it is necessary to develop feed that can reduce fatty liver disease. Therefore, feeding experiments related to this were performed. Experiments 3 and 4 were conducted based on the transcriptome result of experiments 1 and 2, focusing on the nutrition of aged laying hens. The transcriptome result of experiments 1 and 2 showed that laying hens with reduced egg quality, it was confirmed that the expression of genes related to fatty liver disease was high to alleviate fatty liver disease. Experiment 3 was conducted to investigate the effect of dietary concentration of metabolisable energy and neutral detergent fiber on productive performance, egg quality, fatty liver incidence, and hepatic fatty acid metabolism in aged laying hens. At first, fatty liver hemolytic syndrome is a metabolic disease in which excessive fat accumulates in the liver. In particular, fatty liver syndrome related problems are common in laying hens. The picture on the left is what a normal laying hen's liver looks like. However, when fat accumulates within the livers become enlarged and its color becomes lighter. Afterward, if fatty liver disease syndrome worsens further, as you can see, rupture and bleeding occurs, which leads to the deaths of chickens. Therefore, the occurrence of fatty liver disease syndrome in laying hens has negative impact on laying hen productive performance and egg quality, and is a major factor in reducing productive performance of laying hen. The most common way to reduce fatty liver disease is to reduce energy intake. Therefore, it is better to lower the energy content of the feed or add lower energy fiber. This is commonly used method to reduce beta liver. However, to date, research on laying hen relate to the interaction between dietary energy and fiber is insufficient. In addition, the use of lower energy fiber in the feed has been avoided in the past. However, as cost-effective fiber has been receiving attention as having a positive effect on poultry feed, there has been increasing interest in the use of fiber source in poultry feed. Dietary fiber improves poultry's intestinal morphologies, increases nutrient absorption, and improves nutrient digestibility. In addition, part of the fiber plays a role in supplying energy to intestinal bacteria. It also affects the overall intestinal microflora. It also functions to help develop immune system. Therefore, in order to alleviate fatty liver disease, I conducted the following experiment to determine the effect of energy level and fiber content in the feed on productive performance, egg quality, and hepatic fatty acid metabolism in aged laying hens. Material and method of experiment 3, a total of 320 75-week-old highline brown laying hens were allotted to 104 dietary treatments with 8 replicates of 10 hens. The experiment was conducted using a complementary randomized design with 2x2 factorial arrangement. The first factor is AMEN, which consists of normal AMI level and low AMI levels based on the recommendation of the Highline Brown Management Guide. The second factor is NDF, which is also composed of high level and low level. The experiment period was 12 weeks and measurement items were analyzed related to productive performance, egg quality, and liver fat analysis, and hepatic fatty acid metabolism. The following is the composition and nutrient concentration of experimental diets. The low energy and normal energy treatments were formulated with 50 kcal differences. The fiber was adjusted to be byproducts of grains such as wheat bran, DDGS, rice bran, and etc., or feed with high fiber content located on the outside of grain. High NDF feed was formulated by increasing 1.4 times the low-level NDF feed. Productive performance graph according to the dietary AMEN and NDF levels. The x-axis, the treatment group according to dietary AMEN and NDF levels. The y-axis, the numerical value for each item. According to AMEN and NDF levels, there were no significant difference in hen-day egg production and feed conversion ratio between treatments. This is a quality graph according to dietary AMEN and NDF levels. In terms of eggshell thickness, there was an interaction between dietary AMEN levels and NDF levels. The high NDF and normal AMEN treatments show the highest eggshell thickness. Next is main effect. Regardless of the NDF levels, I will talk about the average value of low AMEN and the average value of normal AMN as the main effect. As the main effect, the normal energy group tended to have higher axial strength than low energy group, and high NDF groups tend to be higher axial strength than low NDF groups. In the case of egg yolk color, there was an interaction between dietary AMEN and NDF levels. The egg yolk colors was highest in the high NDF and normal AMEN group. Next is the main effect. Regardless of the NDF level, I will talk about the average value of low AMEN and the average value of normal AMEN as the main effect. In addition, the normal AMEN group has significantly higher yolk color than the low energy group. And high NDF group show significantly higher egg yolk color than the low NDF group. Result is liver fat analysis according to dietary AMN and NDF levels. On the left is subject method about liver color score, and on your right is a liver hematologic score as an indicator of the degree of intrahepatic fats. You can think about the higher the score as the higher the probability of fatty liver disease. There was no significant difference in both the color score and hemorrhagic score in the liver. The results show that the color score and hemorrhagic score were low in the liver high NDF group as compared to low NDF group. That is closer normal liver. Result of measuring the actual liver fat concentration between dietary AMN level and NDF level. The total fat concentration in the liver tended to be low in high NDF and normal AMN group, and the total fat concentration in the liver tended to be high in group low NDF level and low energy group. In addition, normal AMN group had significantly low liver fat concentration than the low AMN group. The high NDF group showed that significantly lower liver fat concentration than the low NDF group. The following are gene expression results related to fat metabolism. This is gene expression graph related to fat metabolism according to dietary and AMN levels. The gene expression of fatty acid oxidation was lower in a low AMN group as compared to the normal AMN group, and fatty acid synthesis show an increase. It appears that this result of gene expression related to liver fat metabolism leads to increased liver fat concentration. The following is a gene expression graph related to fat metabolism according to dietary NDF levels. High NDF groups increase the gene expression of fatty acid oxidation and decrease fatty acid synthesis as compared to the low NDF groups. It appears that this result of gene expression related to liver fat metabolism leads to a decrease in a liver fat concentration. This is the conclusion of experiment 3. Low energy diets showed a negative effect by increasing hepatic fat concentration in aged hens, therefore increasing NDF levels in commercially recommended energy diets of aged hens, decreased hepatic fat concentration, and had a preventative effect on the fatty liver development, and finally improved eggshell thickness. And experiment 4 is effect of dietary supplementation of taurine on productive performance, egg quality, and liver health in aged hens. In experiment 4, we focus on taurine as a functional materials that reduce fatty liver disease. In a previous study on dietary taurine reduced hepatic fat accumulation in broiler chickens. However, there has been little research on a reduction of hepatic fat accumulation by taurine in a laying hen. In particular, it is even more unclear in aged laying hens. Experiment 4 was investigated to determine the effect of dietary supplementation of taurine on productive performance, egg quality, liver fat, and liver health in aged laying hens. At first, taurine is an essential amino acid synthesized from methionine and is the end product of sulfur metabolism. Taurine has many functions such as fatty acid oxidation, neurotransmitter, antioxidation, and immunomodulation. And it has the important function in conjugating bile acid, which plays a role in emulsifying fats. However, the amount of taurine in the major feed ingredient is lacking. In addition, taurine requirement may be increased in stress conditions. It becomes semi-essential. Therefore, in this experiment, a total 320 75-week-old Hyline brown laying hens were randomly allotted to one of 4 dietary treatments with 8 replicates of 10 hens in a 12-feeding trial. The 4 dietary treatments were no taurine, 0.25, 0.5, 1% taurine added. Measurement included productive performance, egg quality, liver fat content, and antioxidant capacity analysis. The following are the productive performance results according to dietary supplementation of taurine. The x-axis each graph is the taurine supplementation level in the feed, and the y-axis is the numerical value for each item. Hen day egg production and feed conversion ratio were not effective by increasing supplementation of taurine in diets. However, increasing supplementation of taurine in diets from 0 to 1% linearly decreased feed intake. The egg weight of hens fed diets containing 1% taurine was less than that those fed basal diets or diets supplemented with 0.25% taurine. The eggshell strength tended to be increased, increasing supplementation of taurine in diets. From this, it could be inferred that as egg weight decreases, eggshell strength increases. Moreover, hens fed diets supplemented with 0.25 or 0.5% taurine show less broken and shellless egg production rate than those fed basal diets or diets supplemented 1% taurine, which showed a quadratic relationship. The following are the results of liver health indicator according to dietary supplementation taurine. Increasing supplementation taurine in diets showed a quadratic relationship for serum gamma-glutamyl transferase concentration with Hens fat diet supplemented with 0.25 or 0.5 taurine, showing less gamma-glutamyl transferase concentration than those fat basal diets or diets supplemented with 1% taurine. Aspartate aminotransferase in blood is also commonly known as unindicated fatty liver. However, serum concentration of aspartate aminotransferase and total fat concentration in the liver were not affected by increasing supplementation of taurine in diets, but decreased numerically. This is the result of antioxidant indicators according to dietary supplementation of taurine. Malondialdehyde concentration in the liver were decreased with increasing supplementation of taurine in diets. The Hens fat diet supplemented with 0.5 or 1% taurine had less malondialdehyde concentration than those fat basal diets or diets supplemented with 0.25 taurine. However, total antioxidant capacity concentration in the liver were not affected by increasing supplementation of taurine in diets. This is the conclusion of experiment 4. Increasing supplementation of taurine in diets of aged laying hens. Decreased egg weight, however, improves eggshell strength and has positive effect on the liver health, in particular by decreasing oxidative stress. And, however, there has little effect on fat accumulation. A summary of today's presentation. Summarized based on the experimental results obtained so far, this study found that the decline in egg quality in hens is also related to liver. As a result, transcriptome analysis, we found that one of the biggest problems for decline in egg quality is related to fatty liver disease. When the interaction between dietary amine levels and NDF levels was investigated in a feeding experiment to alleviate fatty liver disease, as a result, simply lowering the energy content actually worsens fatty liver disease. In addition, increasing high NDF content was effective in reducing fatty liver disease and improve egg quality. Finally, dietary supplementation of taurine improved eggshell strength and showed antioxidant properties. This concludes my presentation. Thank you for your attention.